This is a webinar by Dr. Andres Ursa titled The Importance of the Intestinal Microbiota in the Drug Development Process. It is for the International Scholars webinar to be held in Newcastle in the UK on 28th to the 30th of March 2022. Dr. Ursa has a Bachelor of Medicine and Surgery from the University of Valladolid. He is a Doctor of Medicine and Surgery from Complutense University of Madrid. He is a director of the Clinic and Institute of Integrative Medicine in Valladolid. He is a teaching healthcare official of the regional government of Castilla y León, located in Valladolid. He is a member of the illustrious Ramón y Cajal Academy of Health Sciences. He was awarded the Dr. Gómez Ulla Award for Healthcare Excellence, and he has a care, teaching, and research activity. This is the table of contents of the webinar. Introduction. Since the Human Microbiome Project started in 2007 in the United States, many discoveries have been revealed about the gut microbiota, GM, thanks to existing technology. More than 70 years ago, the interaction between GM and drugs began to be studied. This field was known as pharmacomicrobiomics. MI is increasingly being recognized as an important factor connecting genes, the environment, and the immune system. In addition to the processing of nutrients, intestinal bacteria can intervene in the metabolism of drugs or xenobiotics in general, affecting their absorption and/or toxicity. With microbiome-derived metabolism, drugs can override, inactivate, and decrease their efficacy or convert to a toxic form, contributing to side effects. What has been known as pharmacological idiosyncrasy has to do with the GM that each person who takes the drug has. Due to this, some group of researchers are carrying out studies in this regard to offer drugs on demand in the style of pharmacogenomics, thus contributing to precision medicine. Several authors agree that there has been a significant change in the intestinal microbiota in recent decades, which increases the presence of certain species and reduces others, although the most surprising finding is the loss of microbial diversity observed in developed countries. One of the most important factors involved in the increased use of antibiotics, which reveals important alterations in GM after their use, such as the decrease in phylogenetic diversity and changes in the proportions of the different families. This could alter the development of the immune system and predispose the host to develop bacterial or viral infections, allergies, and alter the metabolism of drugs, as it did several decades ago. Changes in GM configuration can alter host microbial community homeostasis and have health consequences. One of the important activities associated with the GM is the ability to chemically modify the structures of small molecule drugs. The alteration in GM would have repercussions when it comes to interacting drugs with GM by altering its bioavailability. Because of this, it will be necessary to study this issue in greater depth to achieve more effective and safer drugs. Functions of the gut microbiota. Many of the biochemical and physiological characteristics of the host are influenced by the presence of microorganisms in the GI tract. Microorganisms contribute an enormous enzymatic potential in the intestine and perform a wide variety of metabolic functions. There are many bacterial enzymes that catalyze all kinds of reactions. The most common are those that perform reduction and hydrolysis reactions, which can lead to the inactivation of certain compounds, the activation of prodrugs, or the formation of metabolites with toxic effects. Modification of dietary compounds. The gut microbiota transforms inactive dietary compounds into bioactive molecules. Due to this, some soy isoflavones are transformed into compounds with estrogenic activity, such as equal. The microbiota can generate dangerous toxic compounds, such as the formation of trimethylamine from choline and carnitine present in the diet. Trimethylamine is absorbed and converted into trimethylamine oxide in the liver and is a risk factor for cardiovascular disease. The importance of intestinal barrier integrity. The increase in bacterial lipopolysaccharides, BLPS, induced by changes in the GM due to high-fat diets, is characteristic in patients with metabolic diseases such as type 2 diabetes mellitus, DM2, and obesity. This increase in BLPS breaks the normal integrity of the intestinal barrier, increasing its capillary permeability and decreasing BLPS degradation, which leads to the activation of macrophages in the different tissues and generates a low-grade inflammatory state, LGIS. LGIS. And LGIS over time has been associated with obesity, type 2 diabetes mellitus, cardiovascular diseases, and their events. One of the most common side effects of metformin is diarrhea that occurs due to an alteration in GM. We may be treating patients with anti-anorectics, antidepressants, antidiabetics, lipid-lowering drugs, antihypertensive drugs, etc., in the context of an LGIS. Reciprocal influence between drugs and GM. Drugs and GM exhibit a reciprocal interaction. We have an example of this with taking antibiotics. ABs are capable of affecting the structure of endogenous microbial communities by interfering with the synthesis of bacterial nucleic acids or modifying the permeability of the microbial wall. Bacteria, through their enzymatic systems, can act on some drugs and modify their activity. Drug biotransformation by GM. This can be performed directly or indirectly. Direct biotransformation involves a wide variety of microbial enzymes which can modify the drug, which makes it more or less active. Sulfasalazine is activated by GM. Digoxin is inactivated by GM in 5 to 10% of patients taking it. Digoxin transformation by GM. GM reduces the alpha-beta unsaturated lactone ring of digoxin to give dihydrodigoxin. This modification is performed by the intestinal bacterium Egathella lenta, which reduces the binding affinity with the target of digoxin, sodium potassium ATPase, and leads to a loss of efficacy of the drug in the previous percentage. In these cases, the administration of antibiotics achieves a marked increase in the serum concentration of digoxin. Dopamine production by the GM. The intestinal microbial metabolism of levodopa has the capacity to synthesize dopamine. Since peripherally generated dopamine cannot cross the blood-brain barrier, this effect could reduce the amount of levodopa reaching the brain in patients taking this drug. Indirect biotransformation. GM can secrete metabolites that act as a substrate for host enzymes and modify their ability to biotransform drugs. Involvement of drugs eliminated via the bile. Not only drugs administered orally would be affected, but also those purified by the liver and eliminated through the bile. Microbial beta-glucuronidases break down drug glucuronide conjugates and reactivate drugs or their metabolites. A total of 100 drugs have been identified that can be metabolized by beta-glucuronidases in the human gut microbiome. These medications include opiates, estrogens, ibuprofen, midazolam, and their structural analogues. Forms of affectation of a drug to GM. Various chemicals can alter GM directly by changing its composition or indirectly by changing environmental parameters. The use of antimicrobials is the most characteristic example to see their influence on GM. Directly affected. Catecholamines can alter the growth and motility of the IM. This may explain why stress can predispose to infectious diseases. Polyphenols and ethanol can selectively modulate the growth of some commensal bacteria. The use of inhibitors or of gastric acid secretion was related to intestinal infections at the expense of the modification of GM. Bioaccumulation. Some intestinal bacteria modulate the therapeutic effects of drugs through this phenomenon. It happens in duloxetine, rosiglitazone, montelukast, roflumilast, etc. By kidnapping the drug from the bacteria, it limits its bioavailability. A second effect is due to changes in the secretion of metabolites by bacteria. These in turn can produce changes in the composition of the GM, which is usually associated with the onset of side effects such as weight gain due to taking an antidepressant. Influence of the use of antibiotics on the gut microbiota. The effect of an antibiotic on the gut microbiota is influenced by different factors, among which the antimicrobial effect, its mechanism of action, the structure of the microbiota, and the distribution of antibiotic resistance genes in the microbial populations all stand out. Short-term use of probiotics appears safe and effective when used in conjunction with antibiotics in non-immunosuppressed patients. This restores the microbiota if taken correctly. The administration of antibiotics to infants and during childhood alters MI. A high proportion of Enterobacteriaceae and lower proportions of Bifidobacteria, Bacteroides, and Lactobacillus are produced. The effect of ciprofloxacin on GM is marked and rapid, which involves the loss of 1/3 of the taxa and a change in community composition and balance. With the cessation of treatment, the IM begins to return to its initial state. At 6 months, it still shows alteration with taxa that do not fully recover. Modification of the GM in the eradication of Helicobacter pylori. The Helicobacter pylori eradication treatment also eliminates a wide variety of gram-positive and gram-negative bacteria that are part of the commensal microbial community. Triple therapy with both clarithromycin and amoxicillin, together with omeprazole and metronidazole, increased the number of Enterococcus and Enterobacteriaceae with a decrease in aerobic microbiota. Although triple therapy with clarithromycin appears to attain better eradication results, therapy with amoxicillin could be preferable from a microecological point of view, since the qualitative alterations in terms of persistence or appearance of resistant strains appear to be greater in the group treated with clarithromycin. The clarithromycin plus metronidazole regimen shows a drastic decrease in bacterial diversity after 1 week of treatment. Although GM tends towards a pre-treatment composition when antibiotics are discontinued, it remains altered in some cases up to 4 years later. With regard to clindamycin, it was also observed that 2 years after removal of selection pressure, long-term effects on GM still persisted. Antibiotic resistance. Antibiotic-resistant strains can persist long-term in the host, even in the absence of selective pressure. Disturbed GM may be vulnerable to pathogen invasion or growth during its recovery, as nutrients are abundant and resident populations have been reduced. In case of prolonged or repeated antibiotic treatment, it may enable further growth of pathogenic strains, such as Clostridium difficile. GM in patients with diarrhea due to Clostridium difficile. The stools of patients with Clostridium difficile diarrhea are characterized by decreased GM diversity. Almost all antimicrobials have been associated with the appearance of Clostridium difficile, although it occurs more commonly due to the prescription of clindamycin, third-generation cephalosporins, penicillins, and fluoroquinolones. Metagenomics in drug design. The reciprocal interaction between MI and medicinal products is so relevant that the possibility of considering metagenomics when developing a drug and deciding on a therapeutic conduct has been put forth. The intestinal microbiota in obesity and diabetes mellitus. Metabolic endotoxemia mediates a low-grade inflammatory state, LGIS, that activates the immune system and favors proliferation of adipocytes. The generation of active metabolites, such as short-chain fatty acids, SCFA, or bile acids, are involved in anti-inflammatory effects and regulation of hydrocarbon lipid metabolism. The active metabolites induce a reduction in the intestinal gene expression of the adipocyte factor induced by fasting, responsible for inhibiting the activity of lipoprotein lipase in relation to hepatic and adipose storage of fat. There is an intestinal modulation derived from the secretion of peptides, GLP-1, GLP-2, intestinal peptide YY, etc. Gut microbiota in type 2 diabetes mellitus. Deconjugated primary bile acids are metabolized by intestinal bacteria into secondary bile acids, SBA. SBA bind to TGR5 receptors, increasing energy expenditure in muscle, and GLP-1 secretion in L cells of the intestine, improving insulin sensitivity. Colon-mediated induction of inflammation. Lipopolysaccharides, LPS, originate from the outer membrane of gram-negative bacteria and bind to the toll-like receptor 4, TLR4, activating pro-inflammatory pathways that generate a low-grade inflammatory state and decrease insulin sensitivity. Short-chain fatty acids, SCFA, such as butyrate, acetate, and propionate, produced by bacterial fermentation of ingested vegetable fiber, are used as substrates in hepatic gluconeogenesis and lipogenesis, as well as to obtain energy in the colon. SCFA bind to GPR41 and GPR43 receptors with decreased inflammation, increased production of GLP-1 and peptide YY that increase insulin sensitivity. Questions. Does the integrity of the colonic mucosa influence drug metabolism? Does diet affect drug effectiveness by affecting gut bacteria? Is the presence of intestinal dysbiosis taken into account in the participants of clinical trials of antidiabetic drugs? If drug metabolism by GM is found to be deleterious, how can this activity be monitored, both to assess the consequences of metabolism on drug action and to improve patient management? Should we bet more on metagenomics in drug design? Conclusions. Since GM plays important roles in drug metabolism, studies in this regard should be performed with newly designed molecules. As GM changes with age, it should be taken into account when choosing a sample in a drug study. The decreased capacity of the host to metabolize drugs could affect the desired pharmacological action, reducing or prolonging the permanence of the drug in circulation, with possible inefficiency or increased toxicity. Many antibiotics alter IM, so in selecting the sample in a clinical trial, those who have recently taken them should be ruled out. At present, the techniques used in the study of microbiota drug interactions are still largely limited to animal models, which makes it difficult to extrapolate results to humans. However, it seems clear that thanks to the development of new techniques and the increasingly extensive knowledge of the intestinal environment, in the coming years there will be an exponential growth in the number of molecules affected by the intestinal microbiota, as well as confirmation of many hypotheses formulated to date. Future outlook in this field of research points to the use of probiotics as a tool to modify the intestinal microflora to attain greater efficacy and less toxicity from pharmacological treatment. Of all the drugs affected by the human microbiome, probably those with the greatest clinical relevance are those used in the treatment of cancer, as this is a situation in which there is a clear dysbiosis of the intestinal environment, in addition to mostly being drugs with narrow therapeutic margins and multiple adverse reactions. It is necessary to perform further studies to advance in this area of knowledge within the field of clinical pharmacology and to be able to implement this into precision medicine. Thank you very much.